In my research, I analyzed flower tissue data to determine why certain crossbreeds are not able to be pollinated in certain circumstances in tomato plants. And so what I did is I downloaded different tissue sequences from a database, and I analyzed them using two different coding languages, primarily Bash code and an R coding language. And I determined the most upexpressed genes in a successful cross-pollinated species versus an unsuccessful cross to determine the genes that could potentially cause that to happen. In my research, I found that 3 proteinase inhibitors were upexpressed. Proteinase inhibitors are genes that potentially could prevent proteinases from being released in the flower, and I am supposing that that could potentially cause a successful cross-pollination.
My project had to do with amyotrophic lateral sclerosis, which is also known as ALS or Lou Gehrig's disease, and it's a devastating neurodegenerative disease that's characterized by the death of motor neurons in the body. And my research this year actually happened to be a continuation of a project that I conducted last year, and this year I focused on analyzing sequencing data from post-mortem ALS patient spinal tissue using RStudio and R coding language. And my research question was if soluble RNAs in the tissue were enriched for UG sequences. And my results were actually that the insoluble RNAs were rich in UG sequences, which was contrary to my hypothesis, which was that the soluble RNAs would be rich in UG. It has been found to be dysregulated in ALS to see if they were insolubilized or experienced altered solubility in the ALS patient spinal tissues, and my hypothesis was confirmed there. There were pathways that had some altered solubility, and this research is significant because it's one step closer in understanding how ALS functions on a cellular level, and understanding how the disease works on a cellular level will be critical to finding effective treatments in the future.
So essentially, I studied the effects of green infrastructure on the carbon cycle by studying changes in soil microbial function and composition. And so basically, green infrastructure includes things like rainwater harvesting systems, but there are a bunch of different types. So my lab created a GI system in a residential area in Tucson with 4 different basins. Each received a different frequency of water from a different source, and the increased frequency goes from control to passive to active, and then our gray water receives the most water. And my lab sequenced a 16S phylogenetic gene marker from it and plugged that into a program called Fabrotax, which identified the microbes present and their microbial abundance, and then mapped their metabolic pathways. So then I conducted an extensive literature study to try to link the identified microbes to specific functions they've been proven to perform. I focused on 2 functions important for decomposition, cellulolysis and chitinolysis, which are both involved in the breakdown of cell walls. Chitinolysis breaks down chitin and cellulolysis breaks down cellulose. And I actually found that both of those functions increase with an increase in water availability, which suggests that decomposition is also increasing with the implementation of these GI systems, which could have implications for climate because soil releases carbon as it decomposes. So this could have possible impacts on climate change.
So my project explored the diversity and functions of viruses in understudied environments. So viruses have a significant impact on many ecosystems and the organisms within them. So I wanted to learn more about viruses in the understudied environments of symbiotic organisms and also terrestrial environments. And I found that the majority of viruses found with these programs that I used 3 of them, were actually novel, meaning that they're viruses that have not been previously identified before. And that means that they can have significant impacts that we don't know about. So with my research, I want to continue learning about what these viruses do and how they can affect us.
Okay, so I researched if Prosopis glandulosa leaf extract. Prosopis glandulosa, it's a native Sonoran desert plant from here in the border of US and Mexico. And I researched if it could lower the antibiotic resistance of 3 bacteria. The bacteria that I utilized were E. coli, Sarcina lutea, and Staphylococcus epidermidis, while the methods that I utilized were the glycerin extraction method, strict-play method, and the Kuiper-Bauer. And the reason that I did this project is because antibiotic resistance is one of the world's most urgent public health problems. And actually, here in the US, around 2.5 million people get antibiotic resistant infection, while 35,000 people ended up dying. So that was my reason behind doing this project. And the significance is that Well, actually, to my surprise, after doing the research, was that Prosopis glandulosa actually has antimicrobial properties that can lower the antibiotic resistance. And the bacteria that was most susceptible was S. while the antibiotic that had most efficiency was neomycin. And that concluded me that we can possibly make a tablet binded with P. glandulosa.
I designed, fabricated, and engineered a scaffold cuff model, and a scaffold cuff basically promotes the growth and proliferation of stem cells into an implantable organ. And then after I designed, fabricated, and engineered the scaffold cuff model, I tested the different biolinks to see which would promote that stem cell growth the greatest.